Good morning students. Welcome back to P.N.AMERSEY e-learning classes. Students, today we are going to start part 6 of chapter number 9, Reproduction in Animals. Students, this is the last part of this chapter. Our first topic is in vitro fertilization. Now, what is in vitro fertilization? In a healthy woman, the fertilization of egg by the sperm takes place in the oviduct or fallopian tube. The oviducts of some women are blocked due to some reasons. Such women cannot produce babies in the normal way because the eggs released by their ovary cannot meet the sperms and get fertilized in the oviducts. Why? Because the oviducts are blocked. Such women who cannot produce babies are called sterile. Even sterile women can have babies by using the in vitro fertilization technique. In vitro fertilization means in glass fertilization. This is because in this technique fertilization of an egg with sperm is carried out in a glass dish or a glass tube rather than in the body of a woman. This is commonly known as test tube baby technique. In vitro fertilization is known as IVF in short. The in vitro fertilization technique helps the woman with blocked oviducts in having babies as follows. First one is the eggs are removed from the ovary of woman by laparoscopy. In this operation, a small cut is made in the side of woman's abdomen and an optical fiber tube is inserted into her body so that the doctor can see the ovary and take out the tiny eggs. Second step is the woman's husband provides the sperm in the form of semen. Third step is the sperms are mixed with eggs in a glass dish or glass test tube to carry out fertilization. The fertilized egg or zygote develop into embryos. Fourth step is after about a week or more than a week, embryos are placed in the woman's uterus or womb. If the embryos get implanted in uterus successfully, then normal pregnancy occurs and a baby is born after about 9 months. In vitro fertilization or IVF technique is used to help those couples, husband and wives, in having babies who can produce sperms and eggs, but fertilization cannot take place in the woman's body due to blocked oviduct or blocked fallopian tubes. Students, this is full process of in vitro fertilization shown in the form of figures. Now our next topic is viviparous animals. Now what are viviparous animals? Some animals give birth to young ones. So those animals which give birth to young ones or baby animals are called viviparous animals. Vivi means alive, parous means bearing. In viviparous animals, the young one develops in the uterus inside the body of the mother or female parent. When the young one is fully developed, then the mother gives birth, due to which the alive young one or baby animals comes out from the body of the mother. The viviparous animals do not lay eggs outside their body like the birds and reptiles do. In the viviparous animals, the mother gives birth to a fully developed baby. animal called young one. For example, in human beings, the mother gives birth to a baby, so humans are viviparous animals. Some other examples of viviparous animals are cow, dog, cat, lion, tiger, horse, rabbit, rat, elephant, and camel. In fact, all the mammals are viviparous animals. Our next topic is oviparous animals. Now, what are oviparous animals? Some animals lay eggs which later on develop into young ones. Those animals which lay eggs from which young ones or a baby animal are hatched later on are called oviparous animals. Ovi means relating to eggs. Parous means bearing. In oviparous animals, the mother means female parent lay eggs outside its body. The young one of the animal develops inside the egg. When the development of the young one inside the egg is complete, the eggshell breaks open and an alive young one or a baby animal comes out of it. This is called hatching. For example, the hen lays eggs. The young one of hen or chick develops inside the egg. When the development of the chick inside the hen's egg is complete, the eggshell breaks open and an alive chick comes out of it. Since a hen lays eggs from which its young one or a chick are hatched, therefore the hen is an oviparous animal. Some other examples of oviparous animals are sparrow, crow, butterfly, housefly, frog, fish, lizard, snake, ostrich, and crocodile. In fact, all the insects, birds, and reptiles are oviparous animals. It means they are egg-laying animals. Our next topic is metamorphosis. This is a very important topic, students. You must listen to me very carefully. Metamorphosis. We know that a number of animals such as birds, frogs, and insects, etc., are hatched from eggs. For example, chicks are hatched from the egg of hens that I told you just now. Similarly, the frogs and silk moth are also hatched from their eggs. Now, when a chick comes out of the egg of a hen, its overall appearance is almost the same as that of an adult hen, except that it is much smaller than the hen. So when a small chick grows and develops to become an adult hen or cock, then there is not much change in its body appearance. This is the young one of hen or a chick hatched from the egg, and the adult hen looks alike. But this is not so in the case of animals such as frogs and silk moths, etc. For example, the young one, that means larva or tadpole, in frogs hatched from the eggs looks very different from the adult frogs. The process of transformation from an immature form of an animal like larva to its adult form in two or more distinct stages is called metamorphosis. This is the definition of metamorphosis. In most simple words, the change of a larva to an adult animal is called metamorphosis. Metamorphosis occurs in amphibians like frogs and insects such as silk moth, butterfly, housefly, and mosquito, etc. I will explain you the process of metamorphosis with the example of metamorphosis in frog. The hatching of a fertilized egg of frog produces a very immature young one called tadpole, which is a larva. The tadpole or the larva of frog develops gradually and undergoes many drastic changes in appearance before it forms an adult frog. The tadpole looks very different from the adult frog. We say that during its life cycle, a frog undergoes metamorphosis. The transformation of larva into an adult through drastic changes in appearance is called metamorphosis. The change from tadpole to frog is an example of metamorphosis. Students, we can see from this figure which is shown in the slide that a tadpole and the adult frog looks very different from each other. The body features which are present in an adult frog are not present in its immature form called tadpole. Next topic of this video is cloning. Cloning is the production of an exact copy of an animal by means of asexual reproduction. Any two animals which contain exactly the same genes are called genetically identical. An animal which is genetically identical to its parent is called a clone. A clone is an exact copy of its parent. The cloning of a large animal was successfully done for the first time by Ian Wilmot and his colleagues at the Royal Institute in Edinburgh. Edinburgh is in Scotland. They cloned a sheep named Dolly from its parent sheep called Finn Dorset sheep. Finn Dorset sheep is a female sheep. Dolly, the cloned sheep, was born on 5th July 1996. Dolly sheep was the first mammal to be cloned. How the cloning of Dolly sheep was done, I will explain you point-wise. It means step by step. So, cloning of Dolly sheep. The Dolly sheep was cloned in the following way. First step is the normal body cell was removed from the mammary gland of the female Finn Dorset sheep, which was to be cloned. Second step is an unfertilized egg cell was taken from a female Scottish black-faced sheep and its nucleus containing chromosomes were removed, leaving the egg cell empty, having no nucleus. Third step, the nucleus of normal body cell of Finn Dorset sheep was inserted into the empty egg cell of Scottish black-faced sheep. In this way, a new egg cell was obtained, which had the nucleus of Finn Dorset sheep body cell. The new egg cell was implanted in the uterus of another female Scottish black-faced sheep, making it pregnant. And after 148 days, this pregnant Scottish black-faced sheep gave birth to Dolly sheep. Though Dolly sheep was given birth by Scottish black-faced sheep, it was found to be exactly identical to the original Finn Dorset sheep from whose cell nucleus was taken. So, Dolly was a clone of Finn Dorset sheep whose cell nucleus was used in developing it. All the body cells of Dolly sheep contain the same set of chromosomes having exactly the same genes as the fin dorsate sheep cells. So, though Dolly was given birth by a Scottish black-faced sheep, but its real mother was fin dorsate sheep. Since the nucleus from the egg cell is original Scottish black-faced sheep was removed, Dolly did not show any characteristic feature of the Scottish black-faced sheep. For example, the Scottish black-faced sheep had a black face, but Dolly sheep does not have a black face. Students, as you can see in the slide, that first picture shows cloning. In this first picture, A is fin dorsate sheep, B is Scottish black-faced sheep, and C is Dolly sheep. Second picture is of all the steps that how Dolly sheep is produced, and third picture is of Dolly sheep. Students, last topic of this chapter is advantages of cloning. Now, what are advantages of cloning? The technique of cloning enables us to produce exactly identical copies of domestic animals having favorable characteristics. Cloning produces identical copies of the parent domestic animals and helps in preventing desirable features of the parent animals for future generation. For example, first one is a cow which give high yield, high milk yield, can be cloned to produce exactly identical cows which give high milk yield. And second is a sheep which gives high yield of superior quality wool, can be cloned to produce exactly identical sheep which give high yield of superior quality wool. So, the cloning of mammal also has some disadvantages. Many cloned mammals die before birth or die soon after birth. Many times the cloned mammals are found to be born with several abnormalities. This is the end of chapter number 9, Reproduction in Animals. This is the end of semester 1 also. You must listen all the videos of this chapter very carefully, students. This will help you a lot. So thank you, students, and have a nice day.